Okay, and thank you for inviting me to speak about gene editing and PCDH19. This is my first time in this meeting. Okay, so briefly, today CRISPR-Cas9 technology is a powerful approach to perform gene editing. And simply, the introduction of a guide RNA can allow to program the Cas9 to introduce a site-specific double-strand break virtually anywhere in a genome in which where a PAM sequence is located. And the Cas9 cutting is a double-strand blunt end break that occur precisely 3 nucleotides from the PAM sequence. And these double-strand breaks trigger DNA repair process by 2 distinct pathways. The first one is the non-homologous enjoining that joins broken hands and often results in a random small insertion on deletion of some nucleotides introducing premature stop codon. And this mechanism is useful to introduce knockout events. The other one, the second one, is the homology directed repair that through a strand exchange process repairs the DNA damage accurately when a donor sequence containing the correct allele is co-introduced with a CRISPR-Cas9 machine. And this process is very useful to precisely correct the mutation, gene mutation. At Medical Genetics in Siena, we are performing gene editing in several diseases for different genes, Parkinson, Alport syndrome, POMP, and Rett syndrome, and our approach was patented last year. The strategy is based on the design of a vector and the production of adeno-associated viruses that precisely correct the mutation inside cell and in the right tissue, and is based firstly on testing the efficacy in vitro in cell models, so in fibroblasts, iPS-induced pluripotent stem cells, and neurons derived from these cells. And secondly, on testing the efficacy in vivo, for example, in mouse model, or in natural disease dog model, if it exists. For example, in Alport syndrome, there is a natural dog model, so a dog that is affected from Alport syndrome. And through longitudinal patient analysis, useful to identify the exact clinical timing for drug delivery in order to maximize the efficacy of the treatment. And obviously, together with the study mouse model, in order to understand the percentage of correction that is useful to rescue the phenotype. Thanks to a founding of Italian Ministry of University and Research, we recently start the activity regarding the process entitled A Multidisciplinary Approach to Study Protocatery 19 from neuronal function to the cellular interference pathogenic mechanism, where the principal investigator is Maria Passafaro from CNR in Milan. The project aimed to characterize the synaptic function of PCDH19 and to evaluate whether and how the mosaic expression of PCDH19 affects brain circuits in animal model. And this task is led by Maria Passafaro and Maura Francolini from CNR and University of Milan, respectively. The second one task aimed to perform genetic, neuropsychological, and neuroradiological evaluation of patients. This task is performed by Carla Marini's team. And the other one is to generate and characterize the disease-relevant cells from patient-induced pluripotent stem cells and the innovative stem cell from human exfoliate deciduous teeth model, and this task is led by Alfredo Brusco. Finally, our group in Siena and the group of Alfredo Brusco aim to develop and validate two complementary Cas9-based technologies in order to rescue the cellular interference both in vitro and in vivo. All we know that the random X chromosome inactivation leads to mosaic tissue in female with part of the cell express the mutant PCDH19 gene, and the other part express the wild-type form of the gene. And the coexistence of this two cell line is proposed to scramble the cell-to-cell communication. So, as first approach to try to rescue the cellular interference, we plan to generate a homogeneous population of PCDH19 positive cells. And to do this, we plan to perform a Cas9-mediated PCDH19 activation. So this is the situation before the rescue, in which we have an heterogeneous population of PCDH19 positive and negative cells. And we plan to design a guide RNA to target the promoter of the gene, recruiting the acetylation domain, and allow the releasing of the gene from its heterochromatin state. So we plan to validate this approach both in vitro and in vivo, so in human cell lines and in mouse model. And this approach will allow to generate an homogeneous population of PCDH19 positive cells. And this part will be carried out by Alfredo Brusco's team in Turin. As a second approach to try to rescue the cellular interference, we plan to generate a homogeneous population of PCDH19 negative cells, mimicking the healthy males, the mutated healthy males. So this is the situation before rescue. And in this case, we plan to design two guide RNA to program Cas9 that introduce two different, double-strand break, allowing the deletion of the gene by non-homologous end joining repair. Also, this part will be validated in vitro and in vivo, and we plan to generate, with this approach, an homogeneous population of PCDH19 negative cells, and this part will be carried out in Siena. We have already designed our plasmid, contain both guide RNAs and Cas9 for in vitro and in vitro experiment, and our plasmid will be encapsulated in adeno-associated virus in order to deliver the plasmid to the cells. The plasmid also contains a reporter system in order to check for the infection and for the activation and functionality of Cas9. And then we plan to determine the efficiency, of PCDH19 deletion, both at DNA and at protein level, and then to assess whether this deletion is able to rescue the PCDH19 phenotype, both in vitro and in vivo, with the collaboration of the other group involved in this project. At the moment, the in vitro infection in patient-derived cells is ongoing. Specifically, we have fibroblasts from 4 different patients with the listed mutation, and in which we would like to test our approaches. And the IPS generation are ongoing, except for one that we received from Jack Parents, whom I really thank for this. The 3 out of 4 patients have a de novo mutation, and the last is have an inherited mutation from an affected mother. This is the localization of the mutation. We have 3 mutations since 2 patients share the same mutation, that is a recurrent mutation. However, our strategies based on CRISPR-Cas9 mediated deletion, but also the strategies based on Cas9 PCDH19 activation is not mutation dependent. So if both or maybe one of the 2 system works, it could be used for all patients regardless of the mutation. Now I will show you the results that we obtain in Rett syndrome, since this project regarding PCDH19 is at the beginning, start less than 2 months ago. Why, in the other hand, the project in Rett syndrome is at a more advanced stage. Here you can find the fibroblast, patient fibroblast, transfected, so not infected, with different plasmid that are plasmid specific to correct the mutation. Since in Rett syndrome, we would like to correct a precise mutation. And below in the FACS analysis, you can see the percentage of cells in which the Cas9 is active, since the green cells are the cells in which the Cas9 cut, so it is active. These are neurons derived from induced pluripotent stem cell already transfected. Oh. And then we decide to test if different viral serotype could have different efficiency. And here you can see a FACS analysis with negative control, so no-infected cells, and the cells infected by adeno-associated virus 2 and adeno-associated virus 9. So we perform these different infection in fibroblasts, induced pluripotent stem cells, and neurons. And as you can see, in red dashed line, you can see the cells, the positive cells that are also indicate the relative percentage. And these results demonstrate that adeno-associated virus 2 preferentially infect undifferentiated and divided cells, so induced pluripotent stem cell, whereas adeno-associated virus 9 preferentially infect terminally differentiated cells, so fibroblasts and neurons. Given these results, we decide to use adeno-associated virus 9 to infect our terminally differentiated iPS-derived neurons, and this is the fluorescent 48 hours post-transfection. Using next-generation sequencing, we are able to check the correction, to evaluate the correction. And here, in the left panel, you can see the heterozygous non-corrected cells in which you can see the mutated allele, and in the right panel you can see the edited cells in which the mutated allele is less, so the majority of reads are corrected. And the corrected reads have an internal control that is the C. So all the reads that you see here, they have the C are the correct reads, so in which the Cas9 perform the correction. Using next-generation sequencing, we can also quantify the correction. Here you can see the results of correction in fibroblasts in 3 different patients with the same mutation, that is an hotspot mutation. And this is the homology direct repair efficacy, the percentage of efficacy, and also the percentage of the wild-type allele that results after the correction. So, given this, that range from 70% to 98%. So, given these exciting results about gene editing, we will expect to obtain an even more efficient gene deletion, since the gene deletion is more easy to obtain in respect to the gene correction, since it implies a more simple process. This is the group of Siena, and to do these results, I would like to thank also Silvestro Conticello from Florence and Alberto Aricchio from Tigen in Naples. And we recently enrolled a new PhD student dedicated to the PCDH19 project. That is Diana Laverdian. She is here with me in this meeting. And I would like to close my presentation with a special thanks to PCDH19 Hollus for his effort in supporting the research, and all of you for your attention. Attention is up here, but is here.